Hey, everybody, is everybody back from a coffee break? Yeah? Okay, good. So 30 seconds and then we continue. So it will be another interesting session now. Continue with antivirals from James Stewart from University of Liverpool. Then moving to Volker Thiel to research tools from University of Bern. And then closing the session with Susanna Martorell from Zoetis about companion vaccine research. So let's see another round of antivirals. And I learned already that I'm not very expert, but this will be a different mechanism of action, right? No? No? Okay.
Thank you, Zoltan. And before I start, a genuine thank you to the organisers for giving me the opportunity to come here, and the local organisers. This venue is just superb and a great place to hold a workshop. So I'm going to talk about a small part of our work on and I think this was actually Julian's suggestion, to look at molnupiravir in combination with nirmatrelvir, which is the active component of Paxlovid, and also try and introduce immunosuppression for two reasons. You get a more fulminant infection, and also to see if, even in the short term, that affected mutation. Whoa, this is scary. So before I start, I always like to acknowledge who's actually done this work, because as everyone points out to me, I just sit in an office nowadays and drink coffee. So the work was done, and will this actually work? I was given a tutorial beforehand. There we are. We can see that, can't we? So this is my group, all of us here, at the start of COVID. And the work today was done by a very talented group of young scientists, mostly Ellie, Perul and Adam in my lab. The sequence data that I'm going to show was done, I've worked very closely with Julian. That was done by Rebe in Julian's lab, and a huge amount of analysis by her. A lot of the drug work that we've been doing is in collaboration with Andrew Owen, who's also part of the clinical trials network. He's a very talented pharmacologist, and I've learned more in the past couple of years about pharmacology than I would ever know. And of course, Anya seems to collaborate with everybody, and has done a massively excellent job on the pathology of these animals over a number of years. So thank you to all of you. How do I move this forward? Right arrow? Fantastic. So everyone describes themselves as something, and particularly after COVID, I'm not really sure how to describe myself, but so we're predominantly interested in pathogenesis, but over the period of COVID, we've become more actively involved in looking at treatments and drugs, etc. And as part of the pipeline in Liverpool, during the start of the pandemic, I led on developing animal models to be able to, first of all, test a lot of the therapies that were coming through at the beginning, but also to evaluate some of the variants that were coming through. So kind of risk assess, but also look at the biology and pathology and how these viruses behaved as they came through. And so we were lucky enough, my group before that, I should say, was working flu mostly, and a bit of RSV. And we kind of retooled to work on SARS-CoV-2. And we were also fortunate enough to have safety level, BSL, containment level 3 facilities already in Liverpool, including an animal facility that was at BSL 3. So we had all the equipment, which was good. We just had to revise all the risk assessments and get everything up and running, which was not a trivial undertaking for anyone that's thinking of doing it. We in Liverpool, all our facilities are pathogen-free, and we run everything in these individually ventilated cages. And actually for this work, we use cages that are high biosecurity, so they actually have HEPA filters on in and out. So everything's contained. You put this inside a hood to do your work, you bring it back out again. The two models that we set up, a transgenic mouse model, which are based on black 6 mice, but they have the human ACE2 gene introduced under a keratin promoter so that they produce ACE2 in the respiratory tract and other areas. The other model is Syrian golden hamster, which we'd never worked with and has its challenges. But again, the pathogenesis is slightly different in the golden Syrian hamster, so we've worked with the two of them to complement each other over the period of the past few years. So I just wanted to run over these two individual models and the features of them. So as I said, these are based, these transgenic mice are based on BLAC6. They've got the human ACE2 gene. We infect the mice intranasally, so we introduce the virus in the nose. And over the first few days, they appear fairly normal, and then they lose weight rapidly. And weight loss is the only clinical sign that we can find with this virus in mice, but it's very predictable. And in the UK, as in the rest of Europe, but probably not America, for instance, we have a cut-off of 20% weight loss. So anything that goes down towards the 20% weight loss, we have to euthanize. And that's what we use as an endpoint. If we talk about survivability, that's what we use as an endpoint. You get a viral pneumonia, and we can measure either viral load using PCR. We can also look at subgenomic RNA, or we can look at virus titer in the tissues. Obviously, you get a peak around 3 to 5 days in the mice, and then it drops off. But you still get measurable virus at 7 days or so after infection. With this model, the mice don't tend to recover. You can modulate the dose of infection, but it is quite a fulminant model, and mice generally we have to end up euthanizing if they haven't gone far enough. I'm not going to attempt, in the presence of Anya, to talk about pathology, but you do get a viral pneumonia. You can see patches of viral antigen that are present, actually not in the airways, but mostly in the alveoli. And another feature of this model is that you get neurological infection, so you get extra pulmonary infection, somewhat dependent on what strain of virus, but you do get infection of the brain. So it is a model. We've used it a lot in terms of drug development. It's probably a good model for more severe disease in humans, and it does tend to be fatal, if you like. Hamsters, less conventional. So these are an outbred model. They're actually quite hard to work with. As you can see, they don't have a tail, which is where it's convenient to take blood out of, also convenient to put things into. So it's actually challenging to work in hamsters if you're doing drug development, if you want to do PK studies, for instance, or anything like that. However, it is a good model for SARS-CoV-2. Again, we introduce the virus intranasally. You get predictable weight loss, but again, that's the only clinical sign that we can use, weight loss. We've looked at temperature changes, we've looked at behavioural changes, we've looked at other things. There's no real difference apart from weight loss. Again, we can measure viral parameters, and again, we get peaks of viral load, viral replication at around the 3-day mark. This falls off much more rapidly in the hamster, so by day 7, you're not really seeing much virus there. And in contrast, actually, mostly hamsters survive. So if you leave hamsters out beyond day 7, I haven't got this on the graph, but if you leave them out longer, this weight will come up and they survive. So it's actually quite hard to get a hamster to go into that 20% weight loss where you have to euthanize them. In terms of the pathology, again, you get a viral pneumonia. Slightly different pattern here in the hamster where you do get infection around the alveoli, but you also get significant amounts of viral antigen in the airways. And by day 7, you get a lot of consolidation. Viral load's a lot less, so the virus is more or less gone, but you do get this infiltration of macrophages and activation of type 2 alveolar epithelial cells, and considerable consolidation, which, using the morphometry that Anya and her colleagues have very elegantly set up, we can measure the amount of consolidation in the lung, and that's actually quite a useful measure of the success or failure of treatment regimes as well. So just to summarise that, 2 models, slightly different. The K18 mouse, it's actually quite a severe model. Mice are really easy to work with. There's a lot of reagents. They're inbred. They lose weight rapidly. You get these neurological signs, which we're also interested in because of the possibility of using that as a way to get into long COVID. Hamsters, technically a lot more challenging. It's a much less severe model, but there's very limited reagents and you can't get things in and out of a hamster. One thing that I won't show for the benefit of time, but also we've set up transmission models in the hamster. You know, everyone talks about risk of wild animals and things. Pet hamsters, I think, would be really, really not good because they're very, very sensitive and they will transmit very easily from animal to animal. So we've actually developed cages that have a barrier between 2 sides so that we can put an infected hamster on one side and uninfected on the other and look at respiratory drop transmission between the 2. And that's actually quite useful for some drug studies. Some of the compounds that we've looked at, if you do direct challenge intranasally, you don't see much effect. But if you use a transmission model, I think the way the virus is presented is slightly different to the animal, and actually it'll work better. This is just really to demonstrate that you know, over the years we've worked very closely with colleagues in Liverpool, but also we've worked a lot with small pharma and other groups to help them develop compounds, and this is really still ongoing. We've got a couple of projects looking at development of nanobody therapies to try and get more broadly pan-coronavirus reagents, and also looking at drug combinations. So rather than looking at single therapies, we're looking at co-therapies. And also a lot of the data that we generated on variants we fed into places like NervTag, where Julian was, and SAGE as real-time data. So that's just an introduction of the models. So I want now to talk about the experiment where we wanted to look at the effect of drug treatment and the effect of treating with 2 drugs, and in particular molnupiravir and the potential to cause mutation, because there was a lot of talk, and there still is, about how new variants spring up, the involvement of chronic infection, the involvement of people on immunosuppressive therapy, and you know, the role of drugs like molnupiravir in the whole process. So the 2 drugs that we looked at, nirmatrelvir, and I think everyone's introduced this, so I don't know that I need to say much more about it, apart from the fact it targets 3C protease. It's a component of Paxlovid, and Paxlovid is nirmatrelvir plus ritonavir, which alters the PK favourably. The PK of nirmatrelvir isn't particularly good. We haven't used Paxlovid because in the mice, the ritonavir boost doesn't work, so there's no point in having it. You just have to use a large amount of nirmatrelvir because of PK reasons. Second drug, molnupiravir, again, everyone's introduced this. It's a nucleoside analogue, but you do get these transition mutations occurring. And it's debatable as to how fit those mutations are, as we've talked about in previous talks. So this is the scheme that we designed. So the other thing we introduced into this was immunosuppression using cyclophosphamide. And this is an agent, it's a chemotherapy agent, but it wipes out most of your B and T cell response. So that's all taken out of these animals that we treated. And the regime for that is you treat at day -4 and then treat again at day -1. And then actually you remove most of the B and T cell components in these animals. And then we treated in a prophylactic regime with molnupiravir and nirmatrelvir, and then did that. I think actually we did these bi-daily, although I'm just showing one arrow here. Both of these drugs, either singly or in combination, bi-daily. And molnupiravir we used at 100 mg per kg and nirmatrelvir at 500, which is quite a whopping dose. But talking to the pharmacologists who would know, those are human equivalent doses in the mouse because they've done the PK modelling. So groups, we've got vehicle alone, cyclophosphamide alone, molnupiravir, and molnupiravir with cyclophosphamide, nirmatrelvir with cyclophosphamide, and everything together. And we used n equals 4. So this is a very complicated graph, but if I just work through it, vehicle. So I don't think that the mice don't like being given vehicle, which is why they lose a lot of weight initially. And then this kind of comes up and then kind of the virus hits them and they start to lose weight, okay? Cyclophosphamide, you get much more pronounced weight loss with cyclophosphamide, as you might expect. If you give a combination of molnupiravir or cyclophosphamide molnupiravir, actually it's pretty effective. You do get this initial weight loss, and as I said, I think this is because mice don't like being given the drug. But then you get recovery of weight in the face of virus infection with both of those, and also, I should say, with nirmatrelvir. Okay. Again, if we look at survival, and again, the caveat that this is based on 20% weight loss when we have to, by law, euthanize the mice. Cyclophosphamide, actually, the mice drop off pretty quickly in terms of weight loss. Same with vehicle, but actually slightly better. All the drug treatments, actually, all the mice then survive to the end of the experiment. So whatever we do, whether they've got immunosuppression or not, it's actually pretty good. If we look at virology, so either in swabs, nasal tissue or lung tissue, pretty much looks all the same. So this is viral load in swabs, viral load in nasal tissue, and live virus assays in lung tissue. So at day 3 or later on, you get high viral loads with the vehicle, as you'd expect. This drops probably about a log with molnupiravir. You don't seem to get much higher virus with cyclophosphamide, which is, you've still got the innate immunity around, I guess. And then when we're giving the treatments here, molnupiravir again, we're getting about a log difference. Nirmatrelvir is actually much more effective, and combination is more effective than giving single drugs alone, but not at a level that's statistically significant. I think if we upped the numbers of animals in this, that would probably become significant. So I guess the take-home message is that all the drugs are effective, but actually in terms of viral load, given combinations, is probably slightly more effective. And I think that's reflected in the pathology that Anya's done. If you look here, this is viral antigen. With vehicle, you get massive amounts of virus antigen in the lung. Actually, and we don't understand why, you get slightly less virus antigen with cyclophosphamide, and we can't explain that really. Anya thinks it's interesting, but I don't know why. But the drug treatment, you can see here, molnupiravir, Paxlovid's more effective than molnupiravir. Interestingly, I said you get brain infection with this K18 model. All the drugs seem to abrogate brain infection for whatever reason. Now, I'm told by the pharmacologist none of these compounds should actually cross the blood-brain barrier, So it must be hitting the virus hard enough actually in the periphery so that it doesn't get into the nervous system. So this is where Julian's group comes in, and Reby. So we took tissue or nasal swabs, extracted RNA, and sequenced using GridION, and I guess this is midnight amplicon primers to get full genome sequence. Now, because we're doing it under experimental conditions, you can actually, control things a little bit better than human populations. So with all of this, it's very important to know what you're putting in, because then you can understand what's coming out. So we actually sequenced the virus stock that went into the animals so that we could then understand what was happening in terms of changes when it comes out. And as you'll see, it's important to look at minor variants, not just dominant variants. Okay. So if you look at, and this was actually MinION sequencing on here. So there's actually very little coming out in terms of dominant variant changes. I guess it's a short time scale, but I think that's an interesting observation that over one round through an animal, there's not a lot going on. Common observations, aside from what was in the virus stock originally compared to the progenitor, is loss of an amino acid in SP6, but also this mutation in S that's been seen quite commonly, I think, in cats and other animals, and it comes through massively. If you look at this early on, it's very, very rapid, so it's a really strong selection in animals. And if you go back and look at minor variant analysis, this mutation is actually present as a minor variant in the stock. But in tissue culture in humans, I don't think it actually comes through. You put it into mice, it comes straight through. There are a few other mutations, but only in individual samples and nothing consistent. So the answer is there's not a huge amount of dominant mutation going on over the course of the experiment, and that's with any of the treatments, immunosuppression, molnupiravir, anything, which I guess is kind of comforting, I suppose. So then we resequenced, we being Reby, resequenced them with the Illumina, and to look at minor variant analysis, because in spite of what Julian says, I think it's better if you want to actually drill down and get the higher resolution to do Illumina over nanopore. So there's no differences in any of the groups in the transition ratios, but as you'd expect, and as Julian's described, you get a increase in the transition transversion ratios in all the molnupiravir samples. And actually the addition of matravir made no difference to that. So, co-administration of matravir. So if you look here, these are the ratios go up in all those samples. And the other thing we see is not just transition transversions, but just looking at overall levels of mutations, much higher in every case where you've got molnupiravir, and no difference whether these mice are immunosuppressed or not, and also no difference with co-administration with matravir. So in conclusion, both the drugs we looked at are very good at inhibiting viral replication, at least in a therapeutic model. Small benefit if the two in combination. And actually, I think it's been great listening to the talks this morning about the practical application of these drugs in FIP, and I think that's really great work. It's fascinating work. And actually, whether we should be looking at drug combinations, because if you've got a great drug that's tox, you can use less of it if you're using drugs in combination. Little evidence of enhanced mutation. No consistent changes, apart from in the minor variants, we're getting the characteristic mutations with molnupiravir. And that's the lovely waterfront in Liverpool. So if anyone wants to come to Liverpool, we'd love to have you. Thank you very much.
Thank you very much for that. Very, very nice model, actually. I was wondering whether you have looked at long-term infection under immunosuppression without antiviral treatment. What is the development of quasi-species there, or the selection over time? Because after all, that's how Omicron is thought to have arisen from an immunosuppressed patient.
We've tried, sorry, microphone. We've tried very hard to generate long-term infection, and it's actually quite hard. And I think Anya's, with Anya, we've been trying to do this to look at long COVID and the neurological complications that might occur, or other complications that might occur. And, you can, Omicron, the other thing to say with these models, Omicron, is far less pathogenic in hamsters. I mean, almost not, it's not pathogenic in hamsters, right? But it is to a lesser extent in mice. But even with Omicron, if you keep it out long enough, it gets the mice, right? And so they die. And then you lessen the dose down. And we've done a lot of this trying to see how far the dose can go down, and the mice still die. Give them nirmatrelvir at various times. So it's challenging. We've tried very hard, and I agree with you. I think that would be really good. I think there's an alternative hamster model now where they've been managed to do it, but not the golden Syrian, the Russian dwarf hamster. But I don't even want to think about starting another hamster model. But I agree with you, and I think if we can work out a way of keeping the mice going long enough, we just haven't, and I don't think anyone has yet. But I agree with you. I think it's that long-term exposure that's generating these mutants. Yeah.
Any more questions?
First of all, thank you for coming to Cyprus, and thank you for the nice presentation. Maybe I will visit you when I will be in Liverpool. I'm planning to do it this summer. During your presentation, looking on the mice lungs, I was not so sure. I was sure about the case in the hamster, that it was affecting epithelial cells in the lung, but I couldn't figure out from the picture in the mice model was also affecting epithelial cells?
Yeah, both type 1 and type 2 epithelial cells, but it doesn't...
Pneumocytes.
The alveolar epithelial cells, yeah. But it doesn't seem to target the airway epithelia, which is unusual, and I don't understand why.
It's like the ventriloquist in your dummy joke.
Yeah, no, it's just like, did I say that?
George, it's only when you have a mouse-adapted virus, strangely, you can see it in the respiratory epithelial cells as well, like in the hamsters. But the mice, otherwise, the K18, never in any respiratory epithelial cells, just in the nose.
And it's not a, I mean, a lot of people criticise the model because you're artificially driving ACE2, but we don't see a hugely aberrant expression of ACE2. And interestingly, if you, as in Anya, does co-labelling of cells to look for ACE2 and virus infection, you'll find virus in cells that don't contain ACE2, and you'll find lots of ACE2 positive cells that don't contain virus. So I think the infection of different cells is an interesting question, and it isn't just straight down to ACE2 expression. There's more to it. And the neurons, I mean, the neurons have nothing. They have no ACE2. There's, what is it in the...
It's a 10% neuropilin 1.
Yeah, yeah.
Thank you very much.
Thank you very much, James. Nice to see all these antivirals working very well in different models.